Hi students, let us see the concepts of molecular biology today. The topics covered in this molecular biology are given in the slide. Just to introduce about the molecular biology, it is the modern genetics of 19th century. It starts with the genome study. The genome is the complete set of organisms' DNA, whereas chromosome is the specific organized structure in it. Genome contains gene, which is made up of DNA sequences. So the differences are shown here. How molecular biology is emanated from Mendelian genetics. Mendelian genetics dealt with phenotypic expression. It talks about the alleles. Alleles are meant for eukaryotes. For prokaryotes, it is not there. Then from deviations found in the Mendelian law, gave the classical genetics such as recombination, linkage concept, crossing over, and these concepts leads to the production of multiple alleles in eukaryotic organisms. We shall compare the classical genetics and modern genetics. Classical genetics as well as Mendelian genetics dealt with only the phenotypic expression, no genotypic analysis were done. Whereas in modern genetics, the gene level study was done, analyzed at the genetic material, DNA, and the chemical structure of the DNA and their expressions. This modern genetics is modern molecular genetics is otherwise called as molecular biology, which emerged in 19th century, whereas the Mendelian and classical genetics were in 18th century. So this molecular biology is the convergence of different fields such as genetics, physics, structural chemistry to solve the common problem, the nature of the inheritance, structure, chemical substances that make the gene and its actions. There were indirect and direct proof to state that DNA is the genetic material. This inheritance, the root for the inheritance. Indirect evidences are shown here, and the direct evidence came from the experiments. The first experiment was the Griffiths experiment, where the diplococcal pneumonia bacteria of two strains, S and R, 3S and 2R, were injected to mouse. The S could cause the pneumonia and kill the mice, whereas R did not. The S, when it was heat killed, it did not kill the mouse. And when they mixed these two, it could kill the mouse. Thereby, they gave the transforming principle. This transforming principle concluded from the Griffith experiment was that some transforming principle or material or chemical substance might have transferred from the dead S to R and converted the R to S, made it virulent. So the transforming principle was given by the Griffith experiment, but what was the transforming principle was not explained by his experiment. It might be an genetic material from the dead 3S entered live R cells and transformed to 3S. In order to prove what is the makeup of that genetic material, Avery, McLeod, and McCarthy experiment, they have again repeated the same experiment at Griffith, but this time they have isolated the DNA or genetic material alone from the dead 3S and injected to the mouse. That time also it could convert the 2R into 3S and caused the pneumonia, killed the mouse. The same thing they have treated with the different enzymes which can destroy protein, lipid, carbohydrate, and DNA, even RNA. Then they tested in vitro condition. So the observations are shown here, and the same observation was even observed in vivo condition. The mouse also uh, dead when it was treated with the DNA, the enzyme which degrade RNA, protein, lipid, and carbohydrate. Whereas when they treated with the enzyme which degrade the DNA, that could not cause the pneumonia. The same result was further confirmed by another experiment, Hershey and Chase. This experiment is most reliable and acceptable one. They have done the same experiment but in a different condition using a bacteriophage. Two sets of bacteria phage infected cells were grown, and one set the DNA is labeled, radio labeled, whereas in the other set the protein coat of the virus is radio labeled. In the set where the DNA is radio labeled, that DNA when entered into the cells, lysed the cells, and when they separated the virions by centrifugation, they could found the radioactivity at the bottom where the live virus is there. Whereas in the other set, the protein was radio labeled, the DNA was not radio labeled. So there again, the protein was not entering into the cell, DNA only entered and caused the reproduction of the virus. And when they separated in the by centrifugation, the radioactivity was observed in the viral coat in the supernatant, not in the live virions in the bottom. So this confirms that DNA carries genetic information. Now let us see the size of the prokaryotic genome. The genome size and the organization varies between prokaryotes and eukaryotes. Here the size of prokaryotes starts from 0.6 MB to 10 MB. More gene portion, 95 percentage, and the number of genes varies. It commonly, if you look at, they have one chromosome, some may have additional, and some may even have linear. Most of the chromosomes will be circular while some are linear. And in addition, they have the plasmid extrachromosomal DNA. Bacterial genome, if you look at, mostly it will have one chromosome that is called nucleoid. So it is haploid in nature. It is having only one set of gene. Additionally, it will have the plasmids. It may go up to 11. E. coli is having commonly F plasmid. Some are having F plasmid and some are having R plasmid. These plasmids can integrate and multiply along with the chromosome, or else it can multiply uh, self replication mode in an episome manner. And reproduction is done by binary fission. And this DNA, they analyzed the structure. They could find the supercoiling of the circular chromosomal DNA. Supercoiling can happen in right-handed or left-handed manner, positive or negative. Most common is negative. The positive will happen only when overwinding happens. And in underwinding nature, the negative supercoiling happens. Underwinding only happen most of the conditions like replication or transcription. So during that stages, underwinding is more common. So underwinding causes negative supercoiling. So more of negative supercoiling will be there in the chromosome or DNA of prokaryotes. And this genome can be organized in a condensed manner, in a looped manner, or folded form. With the help of other proteins, it can form the scaffold structure. Now, when we talk about the eukaryotic genome, it is complicated than the prokaryotic genome. The size itself is larger. It starts from 12 MB. Prokaryotics below 10 MB. This starts from 12 MB and it is diploid in nature. Two sets of genes will be there, and it is having a mostly linear DNA of duplets in nature, and the gene portion is very very less when compared to the prokaryote. It is only 1 to 2 percentage, and it will have more of the non-gene DNA. So all these non-gene DNA is so condensed and it is kept in the heterochromatic band, whereas the gene DNA will be there in the euchromatin region. The non-gene DNA include intron, regulatory sequences, and non-coding repeats like satellite DNA and so on. And the size of different DNA in different organism is listed in the table. Eukaryotic genome organization itself is very complex. This we call it a supercoiling. In prokaryotes, we have seen the supercoiling positive, negative. Same way, here also the DNA undergo a supercoiling, but here it is around a histone protein. So this will give a basic unit of genome called nucleosome, beads on string. And this nucleosome is formed by the DNA which wound around a histone core octamer. Core octamer means 8 proteins will be there as listed here, and one more histone protein transiently attached also will be there. It is called H1, and the each nucleosome is linked by a linker DNA that is constituting 200 base pair repeat. And the binding affinity, when we talk about the protein, is having all amino acids, but in the binding site, we'll be having lysine and arginine, which are positively charged. The DNA is negatively charged. So these two will attract, and the positive negative attraction will cause this binding. And in the DNA, in the site where the protein bind, will be having more of AT sequence, and it is mostly the minor groove. The complex eukaryotic genome organization, supercoiling again, it will condense as shown in the diagram and arranged as a chromatin and chromosome. The primary structure, if you look at, it is having a covalent backbone structure. You might have studied with the